Hello, and welcome to today's webinar titled Reducing Solvent Consumption with Peptide Easy Clean. My name is Gavin Noble. I'm Process Optimization Chemist at Bachem UK, and I'll be today's moderator. I'm delighted to have with me Robert Zitzbart and Dominic Zarmont from Belyntic, who will be presenting in today's webinar. Just the first point that the first half of the webinar will be pre-recorded, but please submit your questions throughout, and we'll be able to answer them in the Q&A session at the end. Firstly, about Bachem. Who are we? We are a CDMO focused on the production of peptides and oligonucleotides through long-term partnerships with both pharmaceutical and biotech companies. And through this, we contribute to medicines helping people worldwide. We do this through 5 strategic foundations: our people and culture, who we believe really make the difference, and we support people wherever we can. We are a highly technology-based company and believe that this is one of our core strengths and cornerstones of our success. We're aware of our sustainability responsibilities and we take this very seriously. We're a customer-centric business and look to support our customers right through the manufacturing process. And we are industry leaders in operational excellence and quality. And we're able to support our customers right from the research grade through to commercial API supply. And how we do this is seamless transition through our research labs, the CMC development labs, and production of commercialized NCEs and generics. And various departments are able to support these manufacturing. And looking forward, we are expanding our capacity and our capabilities. So in 2021, we were approved capital increase and also expansion of additional manufacturing capacities. By 2024, we will have a new tides manufacturing facility operational, and long term, we're looking for a new manufacturing site in Switzerland. Just to introduce our new Building K, as I mentioned, in 2024, the world's most modern tides fabrication plant. This will allow us to double our manufacturing capability of both peptides and oligonucleotides. It will generate approximately 300 new jobs, and this will be through an investment of 350 million Swiss francs. The core focus of the building will be green chemistry and also implementation of Industry 4.0. So this will allow us to increase the automation, integration of technology, and also use of new and innovative technologies. So how do we drive innovation? One way is through collaboration, particularly with young technology-centric companies such as Belyntic. And the Peptide Easy Clean presented today will offer additional benefits to greener manufacturing of peptides. And we are actively investigating the use of this technology. And proof of our long-term collaboration, this is our 4th webinar today. Yeah. And we're all aware of peptide manufacturing being quite an atom inefficient process and also generating a lot of waste. So how can we reduce this? This is a big focus. One way to increase the or reduce the environmental impact is to investigate alternative solvents, such as binary solvent mixtures that we've presented before, and also in using new technologies such as Peptide Easy Clean, which can help reduce your solvent consumption. And just to cover the topics that will be introduced today. So first of all, the Peptide Easy Clean technology will be introduced. And second, the green solvent use for peptide manufacturing will be investigated. And then thirdly, removal of one particular step that's tricky in peptide synthesis, the precipitation. And also looking at reducing solvent consumption downstream. And so first of all, I would like to introduce Dominic and invite him to take the virtual stage.
Welcome everyone to our webinar about reducing solvent consumption with Peptide Easy Clean, hosted by Bachem. Thank you very much, Gavin, for your nice introduction. My name is Dominic Sharma and I'm co-founder of Belyntic, and I'm happy to introduce you to this webinar and to give you a little introduction to the topic. Now I'd like to start with a little provocative question. Is chemical synthesis of peptides an advantage? Now we all know about the pros of chemical peptide synthesis. It's fast, it's affordable, it's scalable, and it allows flexible chemistry, which is especially important for peptides to turn them from hits to decisive drug leads using, for example, modification chemistry. However, on the con side, we still see the use of hazardous synthesis solvents. The purification requires complex downstream methods as crystallization is rarely possible. And this piles up to a total high amount of waste that's been generated through synthesis and purification. Now, can the peptide industry in future comply with the net zero emission plans? The question is, can we really turn peptide synthesis into a more sustainable, greener solution to comply with these plans? And how will it affect the future of this drug class? Now, we from Belyntic want to tackle this issue and want to contribute with a solution, which comes actually within a single molecule we call the PEC linker that you can see here on the left side. It's a non-hazardous stable powder that allows you to run for the first time universally applicable catch and release methodology for peptide purification and modification. We aim to serve not only R&D but also production scales, so the linker is available in larger scales also for API production. Here you can see the 7 basic steps of the Peptide Easy Clean technology. So in the first step, after solid phase peptide synthesis, you would cap after each amino acid coupling cycle. You couple the PEC linker only to the target peptide in the first step. Then you would cleave off the peptide using TFA as you normally do, precipitate the peptide before you dissolve the peptide. And here in this third step, you have more freedom to, for example, use HFIP or high contents of DMSO to get also complex or difficult to dissolve peptides into dissolution. Then in a fourth step, the catch step applies, in which the oxyamine that's been released after or during the TFA deprotection will react with aldehydes on our solid phase purification media where we use agarose. So in this covalent capture step, only the target peptide is bound through the linker while all truncations remain in solution. And this enables you to wash away the truncations and also other synthetic dirt before an optional sixth step, in which you can apply a modification chemistry because you have an unprotected peptide bound to the solid phase. And then in the last step, you have the safety release, which I will go into more details in this slide. Now, this is the core innovation of our PEC linker and the PEC technology, as we have developed a safety release that actually enables the universal application, and it works like this. So you can see here in green the aryl azide core that's modified with the bromine. Now, in the first step, you would reduce that linker from an azide to an amine, while the linker stays intact through the bromine that sits on the aryl azide core, or now in the aryl amine core. You then need acid to decompose the linker after you have removed all the byproducts from the reduction step. So it's an entirely traceless method. The workup with acids is very easy, and you can choose between whether you want to use high content of TFA, for example, in the subsequent precipitation step, or if you go with low content of TFA or acids and go straight into the lyophilization. You can also vary the acids here and not only use TFA but other acids too, to generate the respective counter ion on your peptide. So again, you have more flexibility with the Peptide Easy Clean technology. Now, here is an example that we've been that we generated together with Bachem some years ago already, but it's still a very decisive example. As you can see here on the left side, the crude peptide of a histone sequence. And it appears clean if you look at the chromatogram, but if you look into the mass, you see lots of truncations that hide beneath the peak. And with our chemoselective method, we can really isolate the target peptide regardless of the complexity of the crude and regardless of the physicochemical properties of co-eluting side products. So PEC is an orthogonal approach to HPLC that enables you to purify and get rid of impurities that are difficult to remove with HPLC. And therefore it's also an ideal complementary system to HPLC to generate high purity peptides. Now we call this PEC grade peptides because you remove all closely eluting impurities, peptidic impurities, and so it's a perfect match for R&D studies in which you want to have high reliability of your assays. And this is given because with this single step purification in a one method fits all protocol that enables you to get a high quality peptide for your assays with high reliability, because you can be sure that you have your peptide, what you want to have, and there are no dirt and there's no dirt hiding beneath your otherwise clean appearing peptide. PEC saves you lots of labor and machinery cost, as it's a very simple method that can be done in simple cartridges, and it's not limited to any choice of peptide length or choice of amino acids composition. With Peptide Easy Clean, we not only provide purification of single peptides, but a bunch of applications are possible or made possible with our technology. So first of all, you can run the purification in parallel in place. So 96 peptide purifications in a single run are no problem. It's a green approach, as on the one hand, as we also show, it works perfectly with greener measures during SPPS, and also it saves a lot of solvents because we do not have a chromatographic separation with a continuous flow of solvents. Something we're going to show today as well is the TFA catch that enables you to completely eliminate the precipitation step after SPPS. And as I also indicated before, you can also run modifications built into this purification strategy to get highly complex peptides in a purified form in a single step. Now, here I would like to cover the three aspects that Robert is going to show you in more details afterwards. First, we look into the synthesis using binary mixtures and greener solvent mixtures for peptide synthesis, and watch out and see if they work nicely with our PEC technology. In the second step of the process, we look at the precipitation that we could eliminate by applying our TFA catch method. In this case, you eliminate the use of dangerous ether, and you also save a lot of time because you just don't have to do the precipitation, lyophilization, and redissolution step. And in the third part, we'll speak about less solvent consumption driven by the PEC purification itself, and how this really comes down to a very, very sustainable solution, not only in R&D, not only in production, but also in R&D, applying the solvent savings for plate formats. So I'm happy to pass on to Robert and wish you all a very nice webinar moving forward. Thank you.
Hello, my name is Robert Zitterbart. Thank you for the Bachem webinar organizers to giving me the opportunity to talk today here in front of you, and also thank you, Gavin, for the kind words. So first I will start off with green solvents and peptide synthesis. So as Dominic has mentioned, our Peptide Easy Clean technology already can have a significant impact on the overall solvent consumption of the peptide manufacturing process. I will guide you through this later as well. But if we start with a conventional peptide synthesis, as you know, it's not very green because the gold standard solvent is DMF. And as such, it's very toxic, for especially reproduction toxic. And this should be replaced, as the EU has recently stated. So usage of DMF after the 31st of December next year will be very complicated. So out of this reason, also out of the reason to have a greener overall process, we also investigated the usage of alternative solvents to DMF. Therefore, we, as a starting point, we took the publications of Bachem and Novo Nordisk, where they investigated the binary solvent mixtures. From this paper, or papers, we took DMSO ethyl acetate as a mixture to being a good alternative to DMF. And we started with DMSO ethyl acetate because we had it in our labs and it's affordable if we convert our own production processes towards DMSO ethyl acetate. What has been described in those publications and also in the nice webinar that was broadcasted in May this year by Bachem was that for the FMOC removal step, with the treatment of 20% piperidine, you need a more polar mixture, so a higher amount of DMSO. And during the coupling steps with amino acid building blocks, less polar mixture is more suitable for giving higher yields. Therefore, we thought that a mixture of DMSO ethyl acetate is nice because you can just vary the ratios, varying from rather higher amounts of DMSO to lower amounts of DMSO, having higher amounts of ethyl acetate. So therefore fine-tuning the different steps of solid phase peptide synthesis. So we took also Bivalirudin as a starting sequences because Bachem, but also others, have used this sequence as a test sequence for testing different solvents and the green SPPS. So in this approach, we use for the FMOC removal DMSO ethyl acetate 6-4, because as I said, higher amounts of DMSO should be beneficial for the FMOC removal. We did it 2 times 5 minutes. In the amino acid dissolution, we also opted for having a rather high concentration of 0.5 molar. And we wanted to have the least polar mixture that still dissolves all amino acids. And what we found was that DMSO ethyl acetate 4-6 was able to dissolve all canonical amino acids to a concentration of 0.5 molar, also showed higher stability upon storage in the freezer, in the fridge. Only FMOC histidine trityl was not dissolved by this mixture, but we used FMOC histidine BOC instead. The DIC was dissolved in pure ethyl acetate. Oxima was also dissolved in DMSO ethyl acetate 4-6. During coupling, then those components were mixed to have a solution of 4 equivalents of amino acids, 5 equivalents of DIC, and 4 equivalents of Oxima, giving this concentrations that are given here. And this resulted in a mixture of DMSO ethyl acetate 3-7. What is a good mixture according also to the Bachem paper in regards of polarity for the coupling step. We did the coupling for 60 minutes. We also had a look in the efficacy of the capping step in DMSO ethyl acetate. DMSO ethyl acetate 1-9 and even pure ethyl acetate works because both acid anhydride and pyridine dissolves well in pure ethyl acetate, and also here in this mixture. And as actually as more unpolar you get, also the efficacy is getting higher. Even though the capping step is very efficient step anyways. We did the washing with DMSO ethyl acetate 228, and between all steps except before capping, because between coupling and capping no washing is needed. And the final wash of isopropanol was performed. So this yielded this crude chromatogram with a purity of the Bivalirudin of 88%. So this was satisfying. The main impurities were here in front of the desired peak, namely truncations and deletion sequences, and also to a minor part spartamide formation. We did this synthesis on PurePep core synthesizer. So this purity of 88%, here is shown again the chromatogram, and again highlighted here are the truncations, and here is aspartamide impurity. Was good because it was even a bit higher than what has been previously reported for DMF, for example. There are in this reported synthesis, purities were around 70%. Crude purity. So, and then we further optimized or further increased the purity from 88 to 93% purity, and removing the truncations and some of the deletions even. But the aspartamide was not removed because our technology cannot remove it. But we could show that the green SPPS using binary solvent mixtures, namely DMSO ethyl acetate, can be used for the synthesis of Bivalirudin, and a subsequent PEG purification can be done without any downsides. So this is a benefit. And therefore you can benefit from solvent savings by using the PEG method. What this really means in an outlook, I also summarized here. It means that an API such as Bivalirudin can be pre-purified by our peptide-based technology, and therefore be highly enriched in content and later on polished by an HPLC. So this means that if you do a single PEG run that is automatable and easy to do, you reduce the salt burden in contrast to crude. You facilitate the dissolution and thus enable a higher loading on smaller HPLC column, in itself already improving the ecological footprint of the method. But now going again back to the SPPS. So we wanted to investigate how generally applicable this DMSO ethyl acetate mixture, this binary mixture is, because as I mentioned, previous reports have concentrated on Bivalirudin or other peptides, but always only single peptides and not a variety of peptides. Therefore, we took 96 peptides that previously have been synthesized in DMF by us, and we did a new synthesis now with binary solvents, namely the ethyl acetate DMSO mixture. The conditions were exactly like I have described before, except at higher cycling, so from amino acid 7 onwards, also one H2 coupling step was added. The peptides had a mean length of 12.4 amino acids, ranging from 8 to 20 amino acids, with all canonical amino acids involved, also cysteines, methionines. The majority of the sequences were derived from SARS-CoV-2, coronavirus peptidome, and mainly spike protein. sequences. The synthesis was done in plate format, as you can see here on the right side, in 10 micromole scale on Ramage resin to get C-terminal amide peptides on an Antavis multipep RSI machine. After linker coupling and TFA cleavage, the peptides were automatically purified by biotage extra hera. Here are the old data, so from the DMF synthesis. Here you see in the crude purities. So here's a box plot representation of the purities. So therefore you see a big distribution of the purities. Here in the box is basically the statistical majority of the purities, ranging around the mean purity of 41.5%. After the PEG purification, there were 85% mean purity, and the majority of the peptides had over 80% purity. So in this context now, it's interesting was a comparison of DMF synthesis against the ethyl acetate DMSO synthesis. Here it showed an improved mean purity, even though here one peptide was not, one peptide more was not detected. Here was one peptide not detected with 0 purity, and here a second one. But sometimes it's even very hard to detect the peptide in when the crude has a very low purity. So here you see after the PEG purification, there was only one peptide where the purity was 0. So this was basically just not there from the beginning. But one of those peptides that were not detected at the beginning actually were there and then had a better purity after the PEG purification. But so the synthesis in DMSO ethyl acetate showed an actually higher mean crude purity, and the purity after PEG was also comparable to the PEG purity before from the synthesis in DMF, so around 85%. The purity distribution was a bit denser here, or more around the mean value of 85% than in contrast to the synthesis in DMF. So let me summarize the first part. So we achieved a nice crude purity of Bivalirudin of 88%. That was further improved by PEG to 93%. This synthesis method we found for Bivalirudin could be also well translated into a synthesis in high throughput format for 96 peptides, improving the crude purity from 42% in DMF to 56%, and this could be further improved by PEG to having a purity of 85%. The aspartamide formation actually was also a bit reduced in DMSO ethyl acetate, as has been reported by Bachem already. So we could confirm that. But a word of warning. So you have to watch out when you use ethyl acetate mixtures, because ethyl acetate is rather volatile. Also, in combination with dimethyl ethyl acetate, it forms an isotropic mixture. So also the DMSO gets evaporated, or the whole mixture gets evaporated. Not so easily, but you have to have a look on that, because especially on this flatbed synthesizer we are using for the high throughput peptide synthesis, you sometimes have vessels that are not sealed or not sealed well, and you have to ensure that it's always sealed well, otherwise they may run dry, or that the concentration in the vessels or the viscosity will change due to the evaporation of ethyl acetate or ethyl acetate DMSO mixtures. The benefits are clearly there. So the DMSO ethyl acetate mixtures They are affordable. They can be easily mixed if you have DMSO ethyl acetate, and they are non-toxic. That's the main thing we do it, right? So for example, you have a very way better feeling if you have to refill the synthesizer and you put like 4 or 6 liters of wash solvents inside, and you don't feel as bad as if you dump 6 liters of DMF in there. So the resulting crude peptides work well with PEG. So there are no drawbacks. Actually, even a bit less side reactions like aspartamide is reduced, so less problems in the PEG purification later on. As an outlook, this is now just been an investigation of 96 peptides, so we have to further investigate this and also others to really tell if this holds true even when more peptides are being investigated. And the mixture itself can also be further fine-tuned. For example, in the FMOC removal solution with 20% of piperidine can be maybe even made more polar. Yeah, something that still needs improvement is to prevent the evaporation of ethyl acetate or ethyl acetate DMSO. So maybe the peptide synthesizer manufacturers have to find solutions if this turns out to be the standard solvent mixture in the future. So now to another aspect that our technology enables and is maybe a potential solvent saver. So now to the TFA catch to eliminate dangerous ether. So ether is usually used, as you know, in peptide production after the TFA cleavage step. So here, after SPPS, we have assembled the peptide here as this purple chain, and then it has to be cleft off of the SPPS resin. Usually TFA is used, here shown as this orange drop. And then to get rid of the TFA, you need ether to precipitate the peptide. And this whole working step is cumbersome and also dangerous because you have to use centrifuges and huge amounts of ether to precipitate the peptide and to get rid of the TFA. We thought, wouldn't it be nice to omit the ether precipitation step to make the process easier and also to save solvents, namely the ether solvent. So going one step back, so how do we do it normally? As I described, so we do the ether precipitation here, and then we do a redissolution of the ether precipitate. This usually works very well. We take DMSO that dissolves almost all peptides, but it may oxidize methionine or cysteine containing peptides. We can also use HFIP, hexafluoroisopropanol, but it's costly and volatile. So it would be better if we can just use the TFA because it dissolves all peptides. So the peptides are already dissolved in TFA during the TFA cleavage. So why should we dump additional solvents inside that we can prevent of use and that are even dangerous in case of ether? So we opted to try a new way of peptide production by using this crude mixture directly in TFA cocktail without any ether precipitation. So avoid the ether precipitation, just go directly to with a dissolved peptide, go directly to the cartridge. So what does it mean again in another drawing? So here it's still the peptide is coupled with our PEG linker. Here's truncation, and if you do the TFA cleavage, it's removed from the SPPS resin. Also the truncation is cleft off, and then what it should do is it should react with aldehydes on the modified PMAs here. So on the filter material, PMA stands for polymethacrylate. So it should form an oxime bond. And the problem is a bit, okay, the TFA is very acidic, and it should also protonate the amino oxy, maybe hampering this reaction. So here we did an investigation with a standard peptide, we call alanine standard peptide. Here this little shoulder is also part of the peptide. This is a CO2 on the tryptophan, giving an artifact shoulder. So here is before immobilization, and this is how it usually looks if you do ether precipitation and then HFIP immobilization. So after 90 minutes, it's completely gone. If you do the same thing in a TFA cocktail with TFA TIS water, after 90 minutes, you see an immobilization of 75%. So this is already good, but it's not, not quantitative yet. And if you leave it longer, then it's also maybe not ideal because you don't want to have your peptides sitting too long in the TFA cleavage cocktail. If you do the ether precipitation and then redissolution in 80% TFA, 20% water, the immobilization gets already a bit better. So the TFA catch works, it does, but can we improve it? So what you can do is, if you have the TFA cocktail, you dilute with water, and this also makes the immobilization quantitative, but it may precipitate some peptides. So this is not ideal. What you can also do is dilute the cocktail with organic solvents, for example, DMF or acetonitrile, to increase the immobilization efficiency. Also, bases can be added, weak bases, to neutralize the TFA. And so 2 examples are shown here. At this time, we use DMF here. We didn't want it to be too green at this point, but it can be also done with acetonitrile in the same way. So if you just dilute the cocktail with DMF and then you do immobilization, and this is the product you get this time now with Bivalirudin, you get a decent purity again, 92%. But not the full yield you get with another method. This you get here, so the peak of the product is higher, and this is actually identical, the peak height, if you do the immobilization with HFIP, so with the quantitative immobilization. But if you neutralize the TFA to some extent, so 7.5 to 15 volume percent of pyridine to neutralize the TFA, and add a little bit of DMF, then you get like the full yield, or the same yield as you would get with a TFA-free immobilization that we know is quantitative. The TFA immobilization is always a bit tricky to analyze. Therefore, we didn't have this comparison here, as I have shown you before. But I can tell you that it got quantitative yield, and when using pyridine and DMF, or acetonitrile can be also used, as I said. So therefore, also we repeated that. Here we took, again, for Bivalirudin, TFA TIS water, 95%, and TIS water each 2.5%, could be directly used for the immobilization, directly meaning that you add the pyridine, you have to cool it a bit, and then dilute it with acetonitrile, and then you do the PEC process. And this gives you a very similar purity and very similar yield compared to the ether precipitated and HFIP immobilized peptide as shown here. So this means, in summary, the TFA catch works, and it can be used to transfer PEC pre-purified peptide directly from SPPS to an HPLC polish without any precipitation step, and without the use of dangerous ether. Because what it means is you take the peptide, cleave it off with TFA, put it on the PEG beads, and then get it out with DMSO water, or get it off with DMSO with acetonitrile water with a bit of TFA. Then you can directly add it to the HPLC for final polish. So the impact is that in the SPPS, also for Bivalirudin, no DMF DCM was required in SPPS. This is compatible with PEG. You can avoid any precipitation step and dissolution step. So this will drastically shorten time to product time and eliminates the dangerous ether. And you have less solvent consumption due to PEG. I will deep dive into this now. So less solvents with PEG purification. Okay, where is this 75% coming from that has been on the slides now already 2 times? So it's coming from a very detailed analysis we did together with Bachem UK, namely with Gavin Noble. So I'm happy to be with him today in the webinar again. So he synthesized this histone sequence very similar to the sequence that Dominic showed. It's just 5 amino acids shorter, but also it's troublesome in purification. So the crude purity was 35%, shown here. A single PEG run could improve it to 80% purity. If you do a single HPLC run with heptafluorobutyric acid, then you can achieve a purity of 57%, but you need a second 2-dimensional HPLC purification with TFA then to have a polish and get 94% purity. With a single PEG run and a HPLC polish, you can get 98% purity. But this is the purities. Now let's have a look on the solvent consumption. So what we compared here is like single reversed phase HPLC purification, as has been done here, the PEG purification, as has been done here, and 2-dimensional HPLC purification, so 2 HPLC runs. Here at the bottom, the solvent consumption of the synthesis, shown in pink, has a kind of big impact. And then here is also the solvents needed for capping and linker coupling. So this has been not included in the HPLC purified samples, and it cannot really be seen because it should be yellow here. That's almost impossible to be seen. So it has a very little impact. What has a bigger impact is the ether precipitation, because in the traditional PEG purification, we do ether precipitation twice. First after the TFA cleavage, and then after the final release. So, and then if you just do the PEG run, so this is here just the green and blue bar, it's very little. And if you compare this to the HPLC purification, a single purification, or double purification, then you see a huge impact. But if you take the overall solvent consumption, you have a reduction of 75% or 85%. And if you now consider this the same thing for 96 peptides, the impact is even bigger. But here has only been calculated the solvent for purification, since we have seen that the impact of capping and also linker coupling is can be neglected. Therefore, there's no difference if you do capping or non-capping, if you do linker coupling or non-linker coupling. Therefore, it can be compared to traditional peptide synthesis and purification. So a manual PEG run and also an automated PEG run in 96 well format consumes around 1 liter of solvents. we ask CDMO, what is the industry standard for purification of 96 peptides? And we were told that an HPLC run of 33 minutes with a 15-minute flow rate is used. And this leads to a consumption of 43 liters per 96 peptides. And also we went into the literature, and there Pentalute has reported usage of a 98-minute gradient for neoantigen library production. And this is in stark contrast to the solvent consumption by our technology. So in contrast to the industry standard, the PEG purification in high throughput format saves 98% solvent consumption, and even 99% as compared to this literature source. Not speaking about the time you save by using PEG, because this you can do in parallel. And as you know, the HPLC purification can be only done sequentially. So let me summarize my part. So PEG offers a greener workflow for peptide manufacturing, especially when you consider greener solvents for SPPS, and the greener solvents also can have a positive impact on the PEG purification. And we offer with a high throughput purification a first-in-class universally applicable parallel purification methodology. And also, I have not mentioned this here, but it has been highlighted in a different webinar with Bachem. PEG can also help to achieve complex cyclic peptides, small proteins, also even proteins with non-canonical amino acids. And therefore, it's very helpful for the assembly of peptide-containing therapeutics and bioconjugates. With this, I give again to Gavin for having the poll questions.
Thank you, Robert, and Dominic also for really, really interesting talks there. And yeah, we'll go on to our poll today. You should see a pop-up on your screen. So what is the criticality of green peptide production in your current operations? So is it business critical? Is it essential for your operations? Is it really a high consideration, something you're going to implement pretty soon? Is it more long-term consideration, or is it not currently consideration? And just wait a few seconds for the answers to come in. Okay. So people are looking to the future here. So one answer of business critical and more of a long-term consideration for most of our attendees today. And so we'll move on to the Q&A section. Few questions to start with, mainly around the green aspects, especially the overall consumption of reagents. So how green is PEG really when you consider everything that you need to use for this process?
Maybe I take that question. So it's always, of course, difficult to define greenness, right? So everyone's talking about green chemistry, and it's difficult to put that in numbers. What we try to follow is the 12 principles of green chemistry, in which, for example, prevention of solvent waste is, for example, one general rule. And I think this is evident from, let's say, the batch process that we have with PEG. It saves a lot of solvent in contrast to HPLC, but also to avoid the use of hazardous and toxic reagents and solvents, and to make lab safety, for example, also more important, like, for example, taking out the ether from the entire process. So it's a combination of several points that we can address using PEG, like reducing solvent consumption, replacing toxic solvents, but also replacing toxic or dangerous components from the process. And also, if you look at the, let's say, energetic consumption of an HPLC, of a large-scale HPLC machine, our system is much more easy, run in a batch reactor that you can use an SPPS reactor, basically. So it's a combination of many, and at least we're trying to approach towards more economically friendly processes in the future.
Okay, great, thanks. So along that thread as well, has the linker been coupled in an automated fashion, and can you use the green solvent mixtures that you mentioned in the talk to do the coupling?
Yeah, I take this question. Yes, actually, funny enough, we even started before knowing about this DMSO ethyl acetate to investigate different solvents for the linker coupling. And it turned out that the DMSO ethyl acetate, especially 1 part DMSO, 9 parts ethyl acetate works especially well for the linker coupling. And this can be also used in an automated fashion. So so basically we have 2 synthesizers in our lab where we do it on a regular basis. So the Intavis multipep RSI that I've shown, there you can just put it in a 50 ml Falcon tube, you can put it inside, and then as a last step of the synthesis, the synthesizer mix together oxymar, the linker, and dipia, and then is adding to the resin. So this is the automated last step for the linker coupling, and this works in a similar way also on gyrus machines. So we have the PurePep Chorus machine in our lab, and this is also using the linker, and you can have 2 modes, whether you have a pre-mix with oxymar or the synthesizer is adding the oxymar. This is further activating the carbonate moiety, and again then adding the base, the dipia, and then it's adding to the resin, and this automatically coupling the linker on the peptidic resin. Does it answer the questions?
Yeah, I think so. So that part is automatable. So theoretically speaking, do you think the whole process could be optimized from start to finish, SPPS right through to PEC, without manual intervention?
Yes, this is. Thank you. Thank you for mentioning this. This is the vision, and it's working. Obviously we are not 100% there, but in theory and from the chemistry, it's all working. So as I just described, so the SPPS is automated, the linker coupling as well. Especially on the gyrus machines, you can also do the automated TFA cleavage. Then you can even have or at this point you would need to do 1 manual step, like with the instrumentation we have right now, to add the TFA solution to the PMA beads, as I have showed, so no ether precipitation needed. Then there's direct immunization, and then also also the whole PEC process can be automated, because it's, as Dominic said, it's from an energetical point of view, it's fairly easy, because it's just adding and sucking and chemicals or solutions, and therefore it's automated, and then you can even have it in a low TFA release. So you have your peptide is released from the purification resin, from the PEC resin, with acetonitrile water, and then you can directly put it automatically on an HPLC for polish, or you have it already pure enough, then you can be automatically frozen maybe and freeze-dried. So yeah, so in theory, the whole process from programming of this sequence to the peptidic powder is generally all automatable, or can be automated. Yeah.
Great. Sounds really interesting. We have a question if you're planning to investigate any other binary solvent mixtures, or do you think this is best left up to other researchers? What are your thoughts here?
Yeah, so these ethyl acetate evaporation is a big problem in the flatbed synthesizer. So actually it caused now a very time crucial synthesis to fail because there was this crash out. So in this regard, actually, we are trying to look in alternatives. I'm a bit, or I like DMSO acetate a lot. It smells well, by the way. And so I'm at this point, I'm not sure if we rather investigate other solvents or rather fix it on in an engineering way to make the sealing better or whatever. But yeah, we at some point we are also investigating other solvents. Yeah.
Okay, thanks. There was another question again around the solvents. There is a worry of hydrogen cyanide generation from oxima.
Yes.
Is this something you're aware of?
Yes, and this is also a good part of the DMSO ethyl acetate as far as I know, because this is something that polypeptide group has seen as well and reported as well. And as far as I know, they have also reported that it's suppressed. The HCN's buildup is suppressed in DMSO ethyl acetate. It's rather a problem in DMF or NMP, but not so much in DMSO ethyl acetate. So another reason for DMSO ethyl acetate, actually.
Maybe if I can add something also to the first question, because what we do is we investigate further the ratio of the two solvents, right? So not so much the solvent itself, but the ratio, because we can fine-tune polarity and therefore enhance or modulate the individual steps during SPPS that might help to prevent side reactions or to improve couplings for specific amino acids or specific sites on the peptide. So that's more the direction we would look into to make a better crude. And that's a very nice thing to do, because you can really fine tune for specific reactions, the polarity, which is one of the key drivers for a good kinetic in the reaction.
Okay, great. So just a final reminder that we have additional webinars available at our Knowledge Center and also on YouTube. And to highlight our publications around green solvent use. And again, there's another webinar on that. And there's more information on our website regarding white paper and also information about becoming a partner with Bachem, whether you're a research grade customer or looking for a fully fledged marketed API. So with that, we'll conclude. Thank you for attending our webinar. Of course, if there are any further questions, please do not hesitate to email in any of this. Goodbye.
Thank you. Bye.
Thank you.